So just as an introduction, we know that the immune system is able to get rid of most primary infections. So primary infections meaning fast exposure with a pathogen to achieve a state of sterilizing immunity. So sterilizing immunity is a situation where the immune system is able to get rid of the infection in the population such that there's no longer transmission of the infection. So this would be an example of where you have fast exposure to an infection, the immune system gets rid of it. You may get exposed again, but you do not get infected, okay? So you don't develop any signs and symptoms of infection or of disease. However, there are exceptions to this. There are some infections in which second exposure, third exposure, one would still get infected and present with signs and symptoms of disease. So this includes pathogens like Mycobacterium, tuberculosis, the HIV, and most of the herpes virus infections. And this is because they have unique, this pathogen is said to have unique factors that would, or other pathogenicity factors that make them, for example, cause long-term infections like your herpes virus infections or HIV, which infects cells of the immune system. Or TB, which causes intracellular infections and has ways in which it evades destruction by the immune system. So the reason that the immune system is able to get to completely clear most of these infections is because of immunologic memory. And you know that immunologic memory is a unique characteristic of the adaptive immune system. So the innate immune system really does not have any memory. A second exposure would just elicit the same type of response as a first exposure. It wouldn't be better, it wouldn't be more effective. Okay, and this picture just shows us the difference between a primary and a secondary immune response. So on first exposure to an antigen, that is the primary, where you get the primary immune response, you do get your, here I'm basically focusing on the adaptive immune response. So for example, an antibody response, as it peaks, you get the pathogen being gotten rid of. And then you do get your antibodies going back almost to baseline. Usually IgM goes back to baseline, which is 0. IgG does not. You do get some memory plasma cells that keep producing IgG. However, on second exposure, you get a peak in the response. So there's an earlier response. The x-axis shows the time. So you can see this peak is more acute compared to this one, meaning the response is quicker, because here you'll be priming the memory cells, whether the memory T cells or memory B cells. And then the production of antibody, for example, will even be greater in magnitude. So the response is larger. And then still the response doesn't go back to baseline, meaning you have an even larger concentration of antigen-specific antibody left in serum. So what has been shown is that after loss of maternal antibody in infants, usually we know that we get passive transfer of IgG from the mother to the fetus, usually beginning in the second trimester, peaking in the third trimester. So you expect a normal infant to be born with maternal IgG. But from about 6 months of age to 9 months, you start getting a decline in this concentration of maternal IgG in the infant as they start producing their own IgG. So it's usually at this point in time where they are most susceptible to infection, because they get, especially if there's a delay, sometimes you get a physiological delay in the infant's own production of IgG. So they tend to get, or we have a state that we call a transient hypogammaglobulinemia of infancy. So in that scenario, at this stage, these infants usually are more predisposed to primary infections because their own immune system hasn't really matured. So in this scenario, vaccination can act as a substitute of primary infection. And that is why you see that most vaccines are given in early childhood. So that would be like the surrogate for primary infection, so that now on second exposure, these children are already protected, they already start forming their own antigen-specific immunoglobulin. So this immunoglobulin that they have would be specific against the vaccine antigens that they've been exposed to, so that now when they later get into contact or get exposed to these pathogens, they do not develop symptomatic infection or disease. So immunization is basically a deliberate stimulation of a host's immune system to protect them from a disease. So this stimulation is basically exposing them, like primary exposure to either primary exposure to a pathogen, or you're giving them the effector products of an immune response against a pathogen. So this is where active immunization and passive immunization come in. So active immunization is where you're administering an immunogen. Usually we'd call that a vaccine, so that the host develops their own immunogen-specific antibodies and immunogen-specific T cells. And then a subsequent encounter would render them protective. Passive immunization is where you're transferring the immune components, okay? And this is still your adaptive immune components. So for your B cells, that would be antibodies. We can also passively transfer cells or lymphocytes, okay? We can also passively transfer immune cell producing tissues. For example, a bone marrow transplant would be transfer of immune cell producing tissues, okay? So the focus of this lecture is going to be on active immunization. We're going to talk further on passive immunization, probably in your fourth year series when we talk about immunotherapy. So active immunization is what we call vaccination, okay? Vaccination is active immunization, okay? So just to mention that passive immunization usually would be given to an individual who has already been exposed, basically in clinical practice, what we call passive immunotherapy, either to reduce immediate risk of infection. So this would be cases, for example, where someone has been maybe exposed to, or let me give the good example of severe SARS-CoV-2 infection or severe COVID-19, where plasma immunotherapy is being given to these severely ill patients. So here we're hoping that you're basically transferring anti-SARS-CoV-2 antibodies in the plasma of someone who's recovered from infection to someone with severe disease, just to help them fight the infection. That's an example of passive immunotherapy. Other examples of passive immunization include what I've alluded to in terms of transfer of maternal IgG from a mother to a fetus. Okay, that would be passive because that means the infant at that point in time in early infancy doesn't have their own IgG, so they're relying on the maternal IgG to be able to fight infection. So for active immunity or vaccination, you do have to expose the host to an antigen, and then you expect them to form their own antigen-specific antibody or antigen-specific T cells. And with that, because you get induction of an adaptive immune response, then you do get development of immunological memory. So the difference between active and passive immunization is that active immunization reduces the risk of future infection, while passive immunization in clinical practice is used to treat current infection, okay? So most vaccines elicit antibodies, some elicit T cell responses. The best would be the ones that elicit both T cell responses and antibodies, okay? And we'll see why in a bit. So how do vaccines work? And this is a picture that just summarizes how we generate an immune response to a vaccine. So you get this an injectable vaccine administered through an intramuscular injection. So you have the vaccine antigen. Sometimes we do need an adjuvant and to just increase the efficacy of that immune response against the antigen. So this is a vaccine that's being administered with an adjuvant. So the adjuvants usually help to increase the antigen presentation because they have the expressed danger signals, also your dumps or your pumps, the pathogen-associated molecular patterns, which increase, and remember that those are what will be recognized by your innate immune cells, like your antigen-presenting cells. So once you've primed your antigen-presenting cells, these are the ones that will activate your adaptive immunity, especially the T-cell response, okay? So your dendritic cells are able to recognize the pumps and dumps within the vaccine, okay? And then be able to take up that vaccine antigen and present it to your both CD4 and CD8 positive T cells. Remember, you can have cross-presentation where you have extracellular antigen being presented to your CD8 positive T cells, okay? Remember your B cells can recognize soluble antigen. So they may still be able to, you may get your vaccine antigen binding directly to the B cell receptor and activating the B cell directly, but that would be a T-independent immune response. However, if you do get, I'm trying to get rid of this thing. If you do get T-cell help or co-stimulation of your B cell with the T cell, then you get a T-dependent response. That's why it's important to have a vaccine that will elicit both a T-cell response and a B cell response, okay? So with T-cell help, you get basically maturation of the antibody response, what you call affinity maturation. So you're getting an antibody whose FAB portion has better affinity to the antigen, okay? And you also get class switching, where you get switching from an IgM, for example, to an IgG response. Remember, IgG would be the long, the antibody with the long half-life, okay? So for your B cell response, you get memory B cell proliferation, as well as memory plasma cells. And the long-lived plasma cells will actually go and niche in the bone marrow. We don't get normal plasma cells in the peripheral blood. So plasma cells don't circulate in blood. They stay in the bone marrow. The long-lived ones, the short ones we may find in the spleen or in the other lymphoid organs like the lymph nodes. For your CD8 T cells, remember CD4 T cells also provide T cell help to CD8 cells by producing cytokines that activate your T cells. And then you do get your antigen-specific effector T cells, CD8 T cells, as well as memory T cells. So for any vaccine immunogen that we're administering to a host, this is the kind of response that we expect, so that we end up with a memory B and a memory T cell response. So what is the effector function of this memory B cells, or rather the antibodies that they produce, as well as the memory T cells? So for the antibodies, the effector functions are the same as we know, as we have discussed before, okay? So these antigen-specific antibodies will perform a neutralizing function, okay? So a neutralizing antibody is one that will prevent the pathogen from binding onto its target cells. So if it cannot bind, it cannot adhere and now colonize and proliferate and then start causing symptoms. For the example of intracellular infections, for example, that caused by virus, let's give the example of SARS-CoV-2. The antibodies, the immunogen is basically the spike protein, which we know for the virus to cause disease, the spike protein has to bind to its host cell receptor, which is the ACE receptor. So a neutralizing antibody will bind to the spike protein and prevent it from attaching to the ACE receptor, and then you don't get internalization of the virus, okay? The other thing that neutralizing antibodies do is that they prevent the actions of toxins. Where you have pathogens that mediate disease by basically production of toxins that now bind to a cognate host cell receptor for the toxin to be active, for example, the cholera toxin, then the neutralizing antibody binds to that toxin and prevents it from binding to its host cell receptor. So that's the basic function of these neutralizing antibodies. And this is what we really expect a successful vaccine to do, to produce neutralizing antibodies. You may get production of other antibodies that are antigen specific against the immunogen in the vaccine, but they do not neutralize. So these ones, we call them binding antibodies. So not all the repertoire of antibody that we produce against a vaccine will be neutralizing antibodies. You can get binding antibodies and neutralizing antibodies. So binding antibodies usually would not prevent infection, okay? So the functional antibody would be the neutralizing antibody. The other things that the antibody can do is activate complement, and we know the downstream effects of that. You get cell lysis, you get an inflammatory response. Antibodies can also opsonize the pathogen, now on second exposure to that pathogen, which basically enhances phagocytosis, and they can also perform what we call antibody-dependent cellular cytotoxicity. For your effector CD8 or cytotoxic T cells that are elicited by vaccines, usually this would be important in intracellular, in the cases of intracellular infections, okay? So they'd be able to inhibit not just viral, but replication of these intracellular pathogens, either by, for viruses that secrete interferons, which we know would cause basically a viral resistance in the neighboring cells. Basically that limits the intracellular entry of extracellular virus into the intracellular space, into uninfected cells. But then the CD8 positive T cells would also kill infected cells, whether they're infected by a virus or a bacterial pathogen. So the effector functions are just the same as we have discussed before. So now we'll just talk about the classification or type of vaccines. This is a very basic or a simple classification where we have the several types under the three main classes. So a vaccine can either be a live vaccine, a killed vaccine, or a subunit vaccine. So the live vaccine usually would be an attenuated human pathogen. So it's been weakened. You don't want to administer a virulent pathogen that would end up causing disease, okay? So the live attenuated vaccines include things like the oral polio vaccines, the measles, mumps, and rubella, and the influenza vaccine. There's one type that is live attenuated. Also the polio, the oral one, is the one that's live attenuated. It could be a non-human pathogen, so a pathogen that causes disease in other animals, but would elicit probably like cross-reactive like antibodies or a cross-reactive T cells because of similarity of that pathogen to the pathogen that causes human disease. So best examples here are smallpox vaccine, where the pathogen that was used as a vaccine is the cowpox, which causes disease in animals, in cows, okay? And also the BCG, we know that the BCG is basically, the component of the vaccine is not really Mycobacterium tuberculosis, but the effector response that you get would be able to limit infection by Mycobacterium tuberculosis, okay? So the BCG is like from the cow pathogen. Then you can have recombinant organisms. These are those ones that have basically been manipulated so that they're not pathogenic. For example, like a vector, either a vector that causes disease that wouldn't proliferate in the human host, okay? The best example I'd give, so here they say experimental, but now we know we have a recombinant vector vaccine against SARS-CoV-2. So the adenovirus SARS-CoV-2 vaccine that uses a viral vector, the chimp adenovirus. So usually it's a virus that would cause adenovirus infection in a chimpanzee. So that is what is being used as the carrier for the SARS-CoV-2 spike protein gene, okay? Killed vaccines are those that have actually been killed. They're not live. They cannot, replicate. Okay, so for example, the pertussis vaccine or the other type of influenza vaccine. And then the subunit vaccines, there are several types, which basically means it's just a certain component. Usually the immunogenic component of the pathogen is the one that is taken and purified and used as the immunogen, okay? So this includes the acellular, pertussis vaccine, toxoids, which are purified toxins, okay, inactivated and purified toxins. For example, diphtheria and tetanus. So this would be relevant for pathogens that mediate disease or the pathogenicity by the toxin that they produce. We have polysaccharide vaccines, for example, the pneumococcal vaccine, the Haemophilus influenza type B vaccine, and meningococcal. And then there are recombinant peptides, okay, like hepatitis B vaccine or human papilloma virus, which are actually like also called virus-like particle vaccines. Okay, so they look like a virus, but they are not. They don't have the genetic component, okay, but the cell surface expresses the immunogen of interest. And then we have DNA and mRNA vaccines. Now we have some that are licensed, not really experimental, like the Pfizer and Moderna mRNA vaccines against SARS-CoV-2. Okay, so we'll describe each of these types of vaccines in a bit more detail, giving the unique characteristics and pros and cons. So the live vaccines were the first to be discovered. If you go and read about the history of vaccination, you'll see how basically we got into development of the smallpox vaccine using the cowpox. You can go and read about what Lady Mary Montagu did and Edward Jenner. So the live vaccines are the most effective, and the best examples are actually smallpox. And remember, the vaccine against smallpox is actually the cowpox as the immunogen. And with this, we've been able to completely eradicate smallpox. We don't get any wild type smallpox infections occurring. I think the last case of smallpox was detected in 1977, and I think that was in Somalia. So since then, there's been no case of smallpox. The only con to this is that there may be some people who are harboring smallpox in some labs, maybe to be used as, warfare, but really there's been no new infection of smallpox since the 70s. The other good example is the oral polio vaccine. There's an injectable polio vaccine that's not as effective as the oral polio vaccine. And as you well know, polio is, or maybe you don't, but polio is basically the next pathogen that people want to get rid of through vaccination. The only problem is that when we get wild type strains, if you're not able to achieve herd immunity, and I'm going to describe herd immunity later, you may get people getting infected by the wild type polio strain. And that is why when we get, especially for countries that are very serious about vaccinating the whole population of children against polio, when we do get scenarios where you're detecting polio infection, it's a notifiable infection, meaning any health center or hospital or clinic where polio is picked up. And it doesn't have to be the actual virus. Cases of acute flaccid paralysis, that has to be notified to the Ministry of Health, and then we get massive vaccine campaigns. I think we had one maybe just like 2 months ago, where we were vaccinating children, even if they've been vaccinated. It's just because you want to ensure that you're completely stopping transmission of polio. So it's the next vaccine that we want to globally, the next disease we want to get rid of globally. And back in the day, there used to be a campaign called Kick Polio Out of Kenya. I'm sure you guys were the children who were being vaccinated at that time. So we still haven't been able to achieve complete elimination, but that's one of the things that is being looked at. There are countries where they're actually not reporting polio infections, like in the US. So in such places, they don't give the oral polio vaccine, they give the injectable one because the risk is much less, okay? So as I mentioned, for the live vaccines, you need to use organisms that are not virulent. You don't want to administer the actual virulent pathogen and end up causing disease, okay? So there weakened such that even if they replicate, they do not cause disease. There are several ways of either weakening them or obtaining non-virulent organisms. For example, one way would be using an organism that actually grows or causes infection in animals, but can produce an immune response in a human host that would be effective against the human pathogen. So the best example of that is using the vaccinia virus. That's the one that causes cowpox, and that has been used as a vaccine for variola that causes smallpox in humans. So the second method of using a non-virulent organism would be weakening the human pathogen. Okay, so weakening it is what we call attenuation. There are several ways of doing it, usually in the lab. So in vitro, for example, using heat or using chemicals or things like that, or genomic manipulation, where you get rid of the pathogenicity factor in that organism. So all this manipulation basically is done in vitro. So an example of that genomic manipulation would be, for example, using viral vectors and ensuring that the pathogenicity factor of that viral vector is rendered inefficient. Okay, and then so now you're using the vector as a transmission pathogen, okay, the one that would get into the human host, and it's going to carry the immunogen of interest. So the best example I'd give is the ChAdOx1 novel coronavirus vaccine, and this is the one we've received here, the AstraZeneca vaccine, which uses, the vector is basically the chimpanzee adenovirus, okay, that is now used to carry the SARS-CoV-2 spike protein gene, okay. So why are live vaccines effective? One, because they replicate, so they can mimic the actual infection by the replication. So you get increase in the dose of the immunogen or of the antigen, because as it replicates, you get the antigen increasing in number. Of course, with better antigen response, if you go back to your lecture on antigenicity, you need an optimal dose, okay? So you're able to sustain an optimal dose that would end up activating your adaptive immune response. The other advantage of live vaccines that they can replicate intracellularly, especially for the live attenuated viral vaccines. It's mimicking an intracellular infection by the pathogen. So the fact that it can replicate intracellularly, you're able to deliver antigenic peptides to MHC class 1. And when you do this, you're able to better stimulate your cytotoxic or CD8 T cells, okay, because you get the MHC class 1 being the one that is delivering the peptide to activate your CD8 positive T cells. The other advantage that they replicate at the anatomic site of infection, which gives a better and more relevant immune response. A good example of this is the oral polio vaccine, which is the one that is live attenuated, versus the injectable polio, which is killed. So injectable polio is given via an intramuscular injection versus the oral polio, which is oral drops. And remember, polio causes a fecal-oral. It's transmitted via fecal-oral route, okay? So the oral polio vaccine mimics the actual infection by the polio virus. So it's going to give a better immune response because you need an immune response within the gastrointestinal system in form of mucosal IgA. So the oral polio would basically give a better relevant immune response as compared to the injectable polio. The other characteristic about live vaccines, and here I think the best example is still the oral polio vaccine, may be excreted and passed on to other unvaccinated individuals. So this is good and it can also be bad. It's good in terms of if you're getting the live vaccine being excreted in feces and you get fecal contamination of food, and that contamination includes the live vaccine, then if someone who is susceptible has not been vaccinated, basically gets exposed via that fecal contamination of food, in some way, well, we don't really know whether they get an optimal dose, but in some way you're actually also reaching them via a vaccination, because they're getting exposed from that fecal contamination, but that contamination contains the live attenuated vaccine. The disadvantage of this is that if that person is susceptible in a way that they're immune deficient, they may be at risk of actually getting the polio infection, okay? And I'll touch on the contraindications of live attenuated vaccines in the context of immune deficiency, okay? The other thing about live vaccines is that the antibodies against many of the live vaccines persist for many decades after infection, okay? So that is a pro. Then if the antibodies persist, then you don't need a booster vaccination. Okay. The attenuated live vaccines can cause serious infections in certain situations. So patients with immunodeficiency, they can actually get infected despite the fact that the pathogen has been weakened. And so because of this, these vaccines are contraindicated in those who are immunodeficient, okay? Whether it's a secondary immunodeficiency or a primary immunodeficiency. So when we go into details of primary immunodeficiencies in fourth year, I'll give you examples of children who have died because of infection caused by live attenuated vaccines, okay? Occasionally, the virus can revert back to its pathogenic form, okay? So it can mutate back and become virulent. And I think this is the risk that they've seen in the US, and that's why in the US, they actually don't administer the oral polio vaccine, which is a live attenuated, because they've seen that the risk of polio is actually low, but then they have a higher risk of, I mean, they're basically just trying to balance the risk of getting oral polio versus the risk of reversion of the pathogen in the vaccine. So they would rather give the injectable form of the vaccine, okay? Okay, killed organisms. So the killed organisms are not as efficient or effective as live vaccines at eliciting a protective immune response, but theoretically they're much safer because you've killed the organism, it cannot revert back to its virulent state, and also it cannot cause infection in an immunodeficient person because it doesn't replicate. So if it doesn't replicate, it also cannot enter the intracellular antigen-presenting pathways, meaning it may not be able to elicit your CD8 T-cell response. Subunit vaccines. Subunits, as I mentioned, are components of the pathogen. So usually the component of interest is the one that is most antigenic, okay? So for example, like some of the subunit vaccines against SARS-CoV-2, the actual, like the mRNA vaccine is a subunit vaccine, but the gene that is used, the mRNA that is incorporated into the vaccine is the one of the spike protein, because the spike protein is the one that is most immunogenic, okay? So subunit vaccines predominantly induce antibody responses, but they can also induce T-cell responses. So in terms of the correlate that we look at to see if one has developed a protective immune response, the easiest to look at would be an antibody response. So usually subunit vaccines are of low immunogenicity if you compare it to the live attenuated vaccine. So these are the ones that would probably need an adjuvant to just boost that immunogenicity so that you're getting a better immune response, okay? The other thing is that the antibody response would be of a relatively short duration if you compare it to the live attenuated, the antibody response of live vaccines. So because the antibody response doesn't last very long, then probably a booster vaccination would be required, okay? So there are various ways of preparing the subunits. One way would be destroying the virulent organism, getting the subunits of interest and purifying it, inactivating it so that it doesn't cause disease. And of course, if you're just taking the subunit, you don't have the whole pathogen, then you're unlikely to actually cause disease with that vaccine. So a good example would be the toxoids, where you actually just take the toxin of the pathogen and purify it so that it doesn't cause disease. It's not able to, you're actually just destroying it so that it's not able, it's still antigenic, but it cannot bind properly to its receptor to cause effector function. Okay, so these are chemically altered toxins. So best examples of toxoid vaccines are tetanus and diphtheria. So they're still antigenic. They can elicit an immune response, but they can't cause disease. And as mentioned, the neutralizing antibodies that are produced would block the effect of the toxin. The other way of preparing subunits is by using recombinant technology. And a good example here is the hepatitis B vaccine. And I guess even the human papillomavirus vaccines. These are virus-like particles. So they actually just take the antigen of interest and grow them in cell culture. So for example, for the hepatitis B, they grow the hepatitis B surface antigen, okay, which is the immunogenic portion, in yeast cells to produce virus-like particles. So you get a particle that doesn't have any genomic, hepatitis genomic material, but it expresses the surface antigen, okay, on the surface of that particle, okay? So here the immune response now produces antibodies against the hepatitis B surface antigen, okay? So this will prevent the actual virus on exposure with hepatitis B. These antibodies will neutralize the virus, okay? Because the antibodies target the antigen that it uses to attach to the liver cells. The other type of subunit vaccine is the polysaccharide vaccines. So this would be, for example, for the capsulated organisms. Remember, the organisms that have a polysaccharide capsule. So, for example, your Haemophilus influenza type B or your strep pneumonia. So the pneumococcal vaccines. So the subunit vaccine just basically is composed of the polysaccharide capsule, okay? So we call them polysaccharide vaccines. These ones are very poor immunogens. If I take you back to your lecture on immunogenicity and antigenicity and the factors that affect that, remember the proteins are better immunogens than polysaccharides. So the polysaccharides usually would elicit a T-independent B cell response. So, you know, they can bind to your B cells and cause an antibody production, but there's no T cell help. But so here then, if you don't have T cell help, remember you don't have affinity maturation, you don't have class switching. So you get a short-lived response. You don't get long-lived memory B cells. You predominantly get an IgM response. You can get an certain subclass of IgG, but it's not going to be a very effective long-term immune response. So to try and overcome this, they've been able to conjugate these vaccines to a peptide antigen. So the peptide antigen, they use various types of peptides. For example, they can use like the tetanus toxoid as a peptide, is that you get both a T cell and a T-dependent B cell response, okay? So this is just a picture showing you the difference between your T-independent response induced by a polysaccharide vaccine versus a T-dependent one that is induced by a conjugate polysaccharide vaccine. So the polysaccharide vaccine is able to cross-link the B cell receptor. Cross-linking means you just get the immunogen binding to more than one B cell receptor, more than one FAB portion. That is what causes the cross-linkage. And then that activates your B cell, is able to differentiate from a plasma cell and produce antibodies. So usually it would only produce IgM and IgG2. You don't get memory, long-term memory B cells being produced. These are short-living plasma cells. So these are not the ones that will go and niche in the bone marrow. This will just be found within the germinal centers of your lymph nodes and your spleen. You don't get affinity maturation. So you're not increasing the quality of antibody production, and you don't get a long-lived response. Okay, and actually it's been shown that infants less than 2 years old may not actually be able to produce a very good immune response to your polysaccharide vaccine. However, when you conjugate it to a carrier protein, so remember your B cell can now, it's an antigen presenting cell, okay? So it can internalize the carrier protein and present that to your, break it to peptide, present it to your T cell. And at the same time, you get this T cell producing cytokines that would activate your B cell. You get co-stimulation, basically. So here is where you now get affinity maturation and isotype switching of your antibody. So you get better class of antibody being produced, better quality, and you get long-lived B cells and long-lived plasma cells. So you get a better immune response also amongst infants, basically. Okay, we'll probably go into a bit more detail of these types of this immunoglobulin isotype and how we use vaccine responses to evaluate the immune status of patients in the setting of immunodeficiency states. The other type of subunit vaccines are the mRNA, or rather the DNA and RNA vaccines. For a long time, they had said, oh, we don't have any of this license for use, but now we do. We have the mRNA vaccines against SARS-CoV-2. So here you just get the gene for the immunogenic protein so only the gene for the immunogenic protein. It doesn't have, these vaccines don't have all the genetic material of the virus. And that is why they are not going to basically transform your cells. The risk that people have talked about of this genetic material inserting into our DNA cannot happen, especially for the mRNA vaccines. So when the mRNA is injected, it just needs to be translated into the protein. Okay, so you get production of the spike protein, which is the immunogenic protein, and then you get an antibody response against that. Okay, so that gene is basically coated onto microspheres, for example, lipid nanoparticles for the mRNA SARS-CoV-2 vaccines. So this results in antibody production. We're not very sure about the T cell response, but I think there's work looking at that. Okay. Okay, so this is just an table just summarizing the types of vaccines. So it's broader, I guess in simplistic form, then the classes would be live, killed, and subunit. And this one just looks at each exact type based on the platform. Okay, here I guess I would call this different vaccine platforms, and it gives examples of the vaccine. So the first one is the live attenuated vaccines. So this include measles, mumps, rubella, yellow fever, influenza, oral polio, and so forth, BCG as well, and varicella zoster for chickenpox. The first live attenuated vaccine used was the vaccinia virus vaccine for smallpox. And you can see where vaccination, I think smallpox, trying to get rid of smallpox basically was where we had really new developments into the science of immunology. It tied in with the studies on vaccination in the 1700s. Then you have the killed whole organism. So here an example would be the whole cell pertussis vaccine, the injectable polio vaccine. Okay, that's the one that's also called Salk. The oral one is the one that's called Sabin. Okay. Rabies vaccine, hepatitis A. The toxoid vaccines are diphtheria and tetanus. The subunit vaccine that basically looks at either a purified protein, a recombinant protein, a polysaccharide, or a peptide. So these are pertussis. So you can see you have different platforms for, like you can have, for one particular pathogen, you can have different platforms, okay? Hepatitis B, which is a virus-like particle. Pneumococcal is a polysaccharide. Haemophilus influenza type B is a polysaccharide. Virus-like particles, like human papilloma virus. Then you have outer membrane vesicle. So here is basically you have like outer membrane of a bacteria with a particular pathogen antigen on the surface of that membrane. So the grouping meningococcal is one example of that. The conjugated polysaccharide, this would be Lipid and pneumococcal. I think those are the ones we have in our setting. Just to note, there's I guess in our market, in the public sector, we have the conjugate polysaccharide strep pneumo vaccine, but we also have the unconjugated one. It's called the polyvalent, I think the 23-valent pneumo vaccine. So that one is not the one we give to children. That one can be given to adults. Okay, the one given to children is the protein polysaccharide one. And remember, I showed you that children under 2 years would not elicit a good immune response against the unconjugated one. The other would be the viral vector. So there's a viral vector vaccine against Ebola, the one that was licensed in 2019, Ebola outbreak. And now we also have the SARS-CoV-2 viral vector vaccine. The nucleic acid vaccines, so the mRNA vaccine against SARS-CoV-2 is a good example of that. And then these other two are still experimental. I guess before last year, the mRNA DNA vaccines would also have been experimental, but now we have licensed ones. Okay, so what's a bit more on adjuvants? Adjuvants are substances that are given, as I mentioned, to enhance the immunogenicity of an antigen. So these ones are given to increase that immune response. So as I mentioned, they provide the pathogen-associated molecular patterns or the damage-associated molecular patterns, which are the ones that drive, recognition by the innate immune system, so that they take up this, they recognize that there's something here, take it up and present that antigen to your T cells. Okay. So live vaccines generally don't require adjuvants because, you're using the actual pathogen, even though it's been weakened. So the pathogen itself is the one providing the pumps. Okay, so these ones are able to stimulate your innate immune cells well, okay? Just like they would in a natural infection. So an example here is BCG. And BCG, as much as it's a vaccine, it's also been used as an adjuvant in some vaccines. So the killed whole vaccines, because it's the whole organism, also are able to elicit your pathogen recognition receptors on your innate immune cells. So, they can act as adjuvants on their own. They really don't need an adjuvant in the vaccine. But the subunit ones, like the recombinant or purified proteins, are the ones that now will need an adjuvant so that you can stimulate your innate immune cells, okay? So usually we have synthetic adjuvants added to them. The most widely used adjuvant is aluminium hydroxide, also called alum. Okay, so it's able to activate macrophages directly. And then this macrophages secrete cytokines, and they're able to also, the cytokines can activate your T cells, and they're also able to present antigen to T cells. Ignore the B cells. We don't get antigen presentation to B cells. So it's thought that alum causes this by causing local tissue damage, okay? Because it's a chemical that you're introducing. It will cause damage to your host cells locally, and then now your damaged host cells will elicit the DAMPs, the damage-associated molecular patterns, okay? There's another adjuvant that has come into the market recently. It's called monophosphoryl lipid. It's derived from the Salmonella polysaccharide, and it's been shown to be able to induce this PRR, the toll-like receptor 4, okay? So what the macrophages do, they now secrete cytokines, which help to upregulate MHC2 class expression, so you get better antigen presentation, okay? And interleukin 12, I don't know that you've covered your hypersensitivity responses. Usually interleukin 12 will skew the immune response towards a T helper 1 response, a pro-inflammatory response, basically. Okay, in terms of accessing the intracellular pathways, I think I've already mentioned this. So the sub, and why it's important, it's because when you do this, then you're able to get antigen presentation by MHC1 and activation of your CD8 T-cell responses. So the subunit vaccines are not able to do this properly. They cannot get into the intracellular antigen processing pathways. And if you don't elicit a cytotoxic T-cell response, then you're not able to get good response against intracellular infections, such as HIV. So I think that's one of the challenges with getting a HIV vaccine. Not only are we not able to elicit the cytotoxic T-cell responses, it's also been difficult to get a good neutralizing antibody response. So remember, for HIV, once one immune cell is infected, that's it. The virus will replicate and produce virions that bad and go out and infect other cells. So if you cannot neutralize it from fast exposure, then it becomes a challenge. So if you had a vaccine that would produce a good neutralizing response as well as CD8, then even if you get some virus bypassing and entering cells, and the CD8 response would get rid of those infected cells. Okay. So there are several ways they've tried to basically enable the vaccine to access the intracellular pathways. So one of this is, using like particles that have lipids. Remember, our cells have a lipid bilayer. So you need to make the vaccine lipophilic so that it can actually gain access into our cells. They would use like lipid micelles so that in the virus-like particles so that they can get into the cells. Another example of this would be the DNA vaccines or mRNA vaccines that have a lipid nanoparticle, okay, like the Pfizer and Moderna vaccine, which enables the vaccine basically to get into the intracellular compartment. The other one is using a vector, okay, that would be able to get inside a cell, okay. Last thing is on just vaccine schedules. Just to basically understand why there are specific vaccine schedules, basically meaning you have certain vaccines that are administered at a certain point in time. So this takes into account the clinical implications of each type of infection. So for example, if you look at the rubella vaccine, the main purpose is to prevent intrauterine infection, okay? Because intrauterine infection with rubella would end up causing deformities in the newborn, okay? So there's no point, there's no much point in giving it before puberty. That's why, for example, for us in our national vaccine schedule, we don't have rubella, we just have measles. But in other countries, or even in the private sector, then some children can get measles, mumps, rubella vaccine. For us, we focus on the measles for the public setting to be administered to infants. Another example is that you have some organisms which can cause very severe infection in infants, especially the capsulated organisms like Haemophilus. So Haemophilus would be, influenza vaccine would be given at that age. And also the strep pneumo vaccine. Before the 2000s, we weren't giving, not even 2000, maybe before 2010, we were not giving strep pneumo vaccine to our infants because the conjugated vaccine wasn't there. Okay, but once that got into the market, then we introduced it into our national schedule. So that schedules can keep changing with new developments because these organisms that cause very severe infection in children under 2 years of age. That's why they have to be administered early. It has to be the conjugated one, okay? The other example is HPV vaccine. We know that the main aim for administering HPV vaccine is to ultimately prevent cervical cancer. So these are administered before sexual debut. So usually at the time of puberty, you don't need to give it in childhood, like to a 6-month-old child. It doesn't make sense, okay? These schedules vary in different parts of the world. So for example, in our setting, measles causes severe infection with severe morbidity. You know the complications of measles. You've covered that in your virology series. So in our setting, measles vaccine is administered in early childhood at 9 months of age, versus in the developed world where, okay, I guess before the anti-vaxxers, I think now they probably see measles because, it got to a time they'd really gotten rid of it. So they stopped giving it in infancy. They were giving it to school-age children because it can spread very quickly amongst school-going children. So that's the difference between developing and developed countries in terms of the measles vaccine. For us, we give it at 9 months of age. But I think they've started seeing a bit of this because of the, anti-vaxxer campaigns. People would say, oh, we've not seen this infection, so why should we vaccinate our kids? Not realizing the reason they have not been seeing it is because of successful previous vaccinations. Okay. So this is just an example. I'll send you this and you can look at the differences between the time of vaccination of certain antigens in the developed and developing world. But I just want to show you what our national schedule looks like. It's called the KEPI schedule. We'll cover more of this in pediatrics. The Kenya Expanded Program on Immunization. So there are certain vaccines, and these are mandatory. The ones in the table are the mandatory ones that have to be given in infancy. And usually the ministry really takes offense when parents refuse to vaccinate your children. It could actually be a criminal offense. So at birth, we administer BCG, oral polio, and hepatitis B. At 6 weeks, there's a combination vaccine of DPT, hepatitis B, and Haemophilus influenza. It's called infant Rixaxa, meaning 6. So these are 6. So these are 5. It's called pentavalent, sorry, because diphtheria, pertussis, tetanus, Haemophilus influenza type B, and hepatitis B. And then they give OPV, pneumococcal, and rotavirus. They repeat this 3 at 10 and 14 weeks. Most of these are subunits. That's why they need the boosters, as you can see. BCG, you can see, is given only once. It's a live viral vaccine. Rota is, I think it's live attenuated, okay? At 6 months, we give vitamin A. This is not a vaccine, but it's been shown to be good at for growth, immunity, and things like that. And at 9 months, we give measles and yellow fever. The ones down here are now like the ones in the private sector. So these are not mandatory. The ones in the table are the ones that are mandatory. As we can see for tetanus toxoid, given to children aged, like school-going children above 7 years, and also pregnant women, because we want to ensure we don't get neonatal tetanus. This is my final slide, just looking at herd immunity. So herd immunity is basically a situation where you vaccinated a certain threshold of the population such that you now prevent transmission to those who are susceptible, okay? So susceptible are those in green. If you don't vaccinate, then almost everyone is susceptible and you can get transmission to them, and some may get the disease, some may die. The threshold for herd immunity is different. It's actually needs to be calculated. So for example, for SARS-CoV-2, we can't really tell you what the threshold is. For measles, I think it's about 80% of the population. Of course, we know we cannot vaccinate 100% of the population, but it's thought that if you get to a certain threshold, then you'll prevent transmission, meaning a vaccinated individual, when they're exposed to the pathogen, you stop transmission at that stage, so they cannot transmit it to a susceptible person. A susceptible person would be maybe a person who cannot get the vaccine because they're immunodeficient or because it's not appropriate for their age. For example, like the SARS-CoV-2 vaccine, we're only vaccinating adults because, one, we haven't studied the vaccine in children. And the reason they didn't focus on children in the clinical trials is because children weren't getting severe disease and dying. Okay, but children can get the infection and they can transmit it. But if you vaccinate a good proportion of adults, then you prevent transmission to children, and then those children can also not transmit it. Okay, so that's basically the gist of your herd immunity. Okay, I will take questions, although we are out of time. Let me see what's on the chat. Is passive immunotherapy the same as plasmapheresis? No, plasmapheresis is basically a technique where you're getting rid of some components or getting some components out of plasma through a machine called an apheresis machine. and then the plasma goes back into the individual who you're getting these components out of. Okay, so can be used, for example, maybe patients with end-stage renal disease, and as an alternative to hemodialysis or peritoneal dialysis, where you just, you'd be getting rid of the uremic component of the plasma, and it goes back to the patient. Can be used, for example, if you want to get some particular cells, like your platelets, for example, from a donor, and then every other component goes back into them, or you just want to get the red cells and things like that. So that is the difference. I think we'll cover more of this in third and fourth year when you're looking at clinical hematology and clinical immunology. Okay, so donation of plasma is not plasmapheresis, basically. Okay, so plasmapheresis is not active. So I just want to basically, I think we're having a problem with the terminology. Pheresis is basically the portion, the procedure where you're filtering someone's plasma through an extracorporeal method. So here the extracorporeal method would be the apheretic machine, and then you're filtering the plasma. Okay, so filtering would be, I guess, like where you're getting certain components of the plasma out. So it's a procedure. Okay, so I guess even the donation of plasma it's not the same thing. I think just go back and look at the terminologies. It's not the same thing as plasma donation. Okay, pheresis, the procedure. Okay, but plasma donation and getting that plasma to infuse into another person is passive immunity. But pheresis is the procedure. Okay. Why is OPV given 3 times yet it is a live vaccine? Probably just to boost the immune response. I think probably when they were looking at the type of immune response they're getting, even if it's a live vaccine, it wasn't as effective as what they expected to give the long-term immunity. Okay, so it's just like the same way we have most SARS-CoV-2 vaccines are given in 2 doses, while you have the J&J one, which is given in 1 dose. So that is informed by studies looking at the immune response in the vaccine recipients. That will determine how many, like the best dosing schedule for a vaccine. Is a hapten the same as an adjuvant? No. So if you go back and look at your notes, a hapten is something that is not able to elicit, and it's not immunogenic on its own. Okay, so it's not the same as an adjuvant. An adjuvant in itself is not immunogenic. It boosts the immunogenicity of an immunogen. So frontline workers need to have some immunizations like Hep B. If this has already been administered in infancy, is it necessary to vaccinate again? No. Probably what needs to be done, I know for us, like when we were in second year, that's when we got our Hep B vaccinations, because we never got it in infancy. So I guess you guys, because you're very young, maybe you got it in infancy. So it will be necessary. One of the things maybe the college needs to do is look at, I don't think the college does this because maybe of cost implications, but I know in the Western world, they look at, like if you've received the Hep B vaccine, they now evaluate you for an antibody response. So if you still have a good antibody response, then you don't need revaccination. However, with Hep B, there are people who are non-responders. So no matter how many times you immunize them, they're not going to have a detectable response. Any other questions or comments?
Yes, I would like to ask. Isn't it that when an effort is involved, we call a procedure active and not passive, like plasmapheresis? There is an effort involved.
Mm-hmm.
to change the plasma of the infected person. Okay.
No, but you see, why we are calling it passive is because the person you're giving the plasma to, you're giving them already formed components. Remember, what you're giving them is antibodies in the plasma, so they're not forming their own antibodies. You're giving them someone else's antibodies. That's why it's passive. Is that clear? When you're talking about active immunization, the host is producing their own antibodies or their own T cells. When you talk about passive, you're giving them already formed antibodies or already formed T cells. That's the difference between passive and active. It's in reference to the recipient. Is that clear? Is that clear?
Yeah, it's clear. It's clear.
Okay. All right. Okay. So CAT is on Monday. It's on HHP 202. I hope you're all registered. It's going to be a 1-hour CAT. I think your class rep had asked me about invigilation. So we've put in measures to just avoid cheating. So you may not have to log on to the Zoom link. The CAT will start immediately at 8 and it will end at 9. If you haven't finished by 9, you're going to be logged off, then your answer is submitted. So just know the timing is going to be stringent. All the best. Have a nice day.
You too.